solidification point. The point at which melted fat regains its original firmness. A fat's solidification point is lower than its melting range, not the same as melting point. To avoid splash burns, food to be fried should be well dried before cooking. Frying, not the cooking method recommended to help prevent heart disease. Butter and lard. Animal fats. Lipid oxidation, a main cause of food spoilage. Atherosclerosis, caused by the buildup of plaque in the arteries. Melting point. One difference between fats and oils. In a laboratory experiment, you should blank. Work in a logical manner, measure accurately, clean up as you work. Mayonnaise is an example of a water and oil emulsion. The sugar found in cow's milk is called blank. Lactose. Insulin. Diabetes is usually caused when the pancreas does not secrete enough blank. A carboxyl group. Fatty acids, like all organic acids, contain blank. Hydrogenation. The commercial process used to permanently change liquid oil to semi-solid fat is called blank. Fats are called unsaturated because of blank the double bonds they contain. Successful frying with fat depends on blank the smoking point of the fat. Rancid. The term used to describe the unpleasant flavors that develop during the oxidation of fats is blank. Is underscore 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 underscore. A foam is a blank dispersion. Gas in liquid. The main function of fat in the body is to blank. Maintain body temperature. 0.3. It is recommended that the percentage of kilocalories a person consumes from dietary fat should be less than underscore 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 underscore. Lipids general groups. 1. Storage lipids. 2. Structural lipids. 3. Others. Storage lipids contains fatty acids and oils, built, stable and easily disassemble. Structural lipids contains phospholipids and sterols, built, provide stability and mobility for membrane components. Other lipids contains cofactors, electron carriers, pigments, hydrophobic anchors, hormones, intracellular messengers, built, provide special functions. Structural groups 1. Backbones, sphingosine and glycerol. 2. Fatty acids, R groups, hydrocarbons. 3. Complex rings, cholesterol, derivatives, cofactors, signaling molecules, vitamins. Storage lipids, fatty acid structure. Hydrocarbon derivatives, carboxylic acids with HC chains, tail to 4 to 36 carbons, may be full or partly saturated, branched or unbranched, may have 3 carbon rings and R groups, methyl hydroxyl, *BioFA not have conjugated double bonds, but usually supported by methylene group. Storage lipids, nomenclature. Based on chain length, double bond character, e.g. palmitic acid, 16:0, no double bonds. Identify position of double bonds, e.g. 20:2, triangle, most common Fa, 12 to 24 carbon unbranched. Chains, even number of carbons, acetate condensation, acetate has 2 carbons. Storage lipids, physical properties. Nonpolar chain, 1. Fa poorly water soluble, 2. Slight solubility B, C carboxyl group, or hydroxyl. Her side chain. Storage lipids, physical properties. Room temperature, 1. Saturated Fa or waxy, 2. Unsaturated Fa or oily, temperature outside. Changes, body tries to adjust by changing fluidity. Saturation. Saturated chains are linear, desaturated chains contain bends. Triacylglycerols. Fa esters of glycerols, cleaved by LIPASES, hydrolyze ester linkages. Triacylglycerols, qualities. 1. Hydrophobic, 2. Nonpolar, 3. Nearly insoluble in water, serve as storage forms of metabolic fuel, energy. Triacylglycerols, structures. Glycerol backbone, 3 fatty acids, ester bonds. Mixed triacylglycerols. 2 or more different Fa. Simple triacylglycerols. Only one type of Fa. Triacylglycerols, advantages fuels. 1. Hydrophobic, no need for hydration, solvation layer, saves energy. 2. Carbon atoms highly reduced, yield greater than energy than carbs through oxidation, catabolic pathways, e.g. 15 mg of triacylglycerols equals 1350 mg of glycogen. Waxes. Esters, 1. Long chain, 14 to 36 C, saturated, unsaturated Fa, 2. Long chain, 16 to 30 C, alcohols, melting points higher than triacylglycerols, waterproofers for feathers, prevent evaporation from holly, rhododendron, poison ivy, polishes and lotions, derived from PUM, lanolin, sheep's wool, beeswax. Membrane lipids, 3 general classes. 1. Glycerophospholipids, 2. Sphingolipids, Glycerophospholipids. 2 fatty acids joined to glycerol backbone, EC1 fatty acid, C2 fatty acid, C3PO4 alcohol. Sphingolipids. 1 Fa, 1 fatty amine. Sterols. rigid core of 4 fused HC rings. Phospholipids. Some glycerophospholipids and sphingolipids. 1. Polar head group. 2. Fa. Glycolipids. Some sphingolipids with a simple sugar instead of phosphate. Sphingolipids. Sphingosine backbone, C1PO4 choline, C2FA, no C3. Sphingolipids. Sphingosine backbone, C1 mono or oligosaccharide, C2 fatty acid, no C3. Glycerophospholipids. Derivatives of phosphatidic acid. Glycerophospholipids. Structure. Glycerol backbone, C1 fatty acid, C2 fatty acid, C3 phosphate polar head group, alcohol. Asterisk, glycerol is not chirol, asterisk, but glycerol phosphate is chirol. Ether lipids. Phospholipids with Fa and ether linkages, I, plasmalogen or platelet activating factor, tilde 50% of. Hard phospholipids are plasmalogens, functional significance of ether, not ester, linkage not known, maybe function in membrane trafficking, anti-war agents, no strong evidence of role in health. Sphingolipids. Structure. Derived from sphingosine, 3-carbon backbone, sphingosine long chain Fa polar head group. Head group joined by phosphodiester her glycosidoc linkage, CERAMIDE, Fa attached to amide at C2. Parent sphingolipid. Sphingolipid function. Cell recognition molecules, especially in neurons, blood group determinants. Three classes of sphingolipids differ in head groups: 1. Sphingomyelins, 2. Glycosphingolipids, 3. Gangliosides. Sphingomyelins. Head group equals phosphacholine or phosphethanolamine, asterisk, therefore, they are PHOS-PHOLIPIDs. Similar to phosphatidylcholine, asterisk, no net charge on head group at physiological pH, asterisk, often called glycosphingolipids. Mostly on outer side of plasma membrane, head groups have one or more sugars on C1 of ceramide. CEREBROSIDES, one sugar on ceramide, GLOBOSIDES, more than one sugar on ceramide, asterisk, no net charge at pH 7.0 equals neutral glycolipids. Sphingolipids, clinical, forensic significance. Glycosphingolipids, blood group antigens. Sphingolipids and disease. Generalized gangliosides, Tay-Sachs disease, Gaucher's disease, Niemann-Pick disease, Fabry's disease, Sandhoff's disease. Gangliosides. Polar head groups equals oligosaccharides, one or more N-acetylnuruminic acids, sialic acid, asterisk. Terminal sugars on a lot of carbs. Galactolipids. Different from glycosphingolipids, no nitrogen, no phosphate, plants conserve phosphate. Galactolipids. Lipids with a lot of galactose, different from Glycosphingolipids, no nitrogen, no phosphate. Plants conserve phosphate, located in thylakoid membranes of chloroplasts, most abundant membrane lipids on Earth 1,2-diglycerol 1 or 2 galactose at C3 *, no phosphate. Not normally abundant in soil. Sulfolipids 1,2-diacylglycerol sulfonated glucose. Sulfate can act like phosphate, chemically. Degradation of phospholipids and sphingolipids. If signaling products from cleave of certain bonds by specific enzymes, membrane lipids equals greater than signaling molecules. Phospholipids cleave phospholipids. 1. First product is lysophospholiPid. Functions in signaling. 2. Second product is fatty acid. Sterol structure. 4 fused carbon rings. Sterols not synthesized by bacteria. Sterol nucleus. 1, 3, 6 carbon rings. 2, 1, 5 carbon ring. 3 nearly planar, made from simple precursor acetate CH3 2 carbons. Sterol function. 1. Passive role. 2. Active role. Sterol, passive role. Structural lipids in nearly all cells but bacteria. Cholesterol is amphipathic, polar head group, nonpolar tail, alters membrane fluidity and assists in lipid transport. Low temp prevents HC crystallization. High temp decreases overall fluidity. Sterol active roles. Diverse roles. 1. Steroid hormones, potent signals for gene expression. 2. Bile acids, derived from cholesterol, emulsify fats to make them accessible for degradation. 3. Precursors, vitamins and hormones. Lipids, signals, cofactors and pigments. Phosphatidylinositols. Intracellular signals and cofactors. Function, second messengers. A signal induces activation of reactions greater than produce other molecules and amplify initial signal, other molecules, or second messengers. Amplification may be in several steps and it is part of signal transduction pathway. Phosphatidylinositol, general, reside in plasma membrane, can become phosphorylated in several places. Derivatives are second messengers. Phosphatidylinositol 4,5-biphosphate. Lipid on inner surface of plasma membrane. Functions. 1. Bind cytoskeletal proteins. 2. The bind specific soluble proteins to promote membrane fusion. 3. Bind second messengers after receptor-ligand interactions. Second messengers. 1. IP3, 2. Calcium, 3. Diacylglycerols, PIP2 and IP3. PIP2 equals greater than inositol 1, 4, 5 triphosphate diacylglycerol. Second messenger, causes release of intracellular calcium from ER. Calcium is a second messenger and COFACTOR for protein KINASEC, float in cytosol, interact in HER, activates ATPase. Calcium goes into cytosol. Diacylglycerol. Second messenger, cofactor for protein kinase C. When protein kinase C becomes active, it phosphorylates proteins. Need two of these. Part of one signaling pathway. Phosphatidylinositol ATP equals greater than phosphatidylinositol 4, 5-biphosphate 1 equals greater than inositol 1, 4, 5-triphosphate equals greater than relase of intracellular circuit 2 equals greater than regulation of other enzymes by circuit 2. Also activate protein kinase C. 2. Icosanoids. Intercellular messengers. Paracrine hormones act near where released. Diverse functions. Derivatives of arachidonic acid. 20 carbons. 3 defined classes, aka structural classes, each one centered on particular function. Icosanoids. Diverse functions. 1. Inflammation. 2. Pain response. 3. Blood clot formation. 3. Fever. 4. Regulation of BP. 5. Gastric acid release. Classes of icosanoids. 1. Prostaglandins. 2. Thromboxanes. 3. Prostaglandins. 1. 5-carbon ring. 2 groups with many subtypes. 1. PGE equals ether soluble. 2. PGF equals phosphate. Water soluble. Regulate CAMP synthesis through adenylate cyclase. CAMP. 1. Second messenger. 2. Cofactor. Thromboxanes. 1. 6-carbon ring, produced by platelets, involved in platelet-derived activities like clotting. Synthesis inhibited by nonsteroidal anti-inflammatory drugs, ibuprofen, aspirin, acetaminophen, etc. Inhibit clotting associated with thrombus that would cause heart attack. Cyclooxygenase make HTE rings 3-membered ring. Leukotrienes contain 3 conjugated double bonds, open, no cyclic structure, no rings, serve as signaling molecules. Overproduction causes asthma attacks and equals greater than anaphylactic shock, WBC, very key B.C. of immune function, made by live cyclooxygenases. Nonsteroidal antiflammatory cells inhibit prostaglandins. Steroid hormones, messengers B, W tissues. Oxidized derivatives of sterol, sterol nucleus but no alkyl chain, move through bloodstream as a complex with proteins, bind to highly specific receptors in nucleus, bind with high affinity. Low concentrations have max effects, induce gene expression and changes in metabolism. Steroid functions. 1. Sex hormones. 2. Anti-inflammatory drugs. 3. Treat asthma and arthritis. Hormone precursors. 1. Vitamin A. 2. Vitamin D. Vitamin A. Derivatives are metabolic and cell regulators. Retinoic acid regulates epithelial cell gene expression through nuclear receptors. Retinal, photosensitive pigment in rod and cone cells. Produces a neuronal response. Precursor is B-carotene. Deficiencies equals greater than skin and eye disorders, wrinkles and night blindness. Vitamin A. Essential for site oxidation of alcohol to aldehyde, vitamin A, retinol, equals greater than 11 CI's retinol. Visual pigment, ring and hydrocarbon, visible light turns into trans equals greater than neuronal signal to brain, oxidation of retinol aldehyde to acid equals greater than retinoic acid equals greater than hormone, dietary, B, carotene, equals greater than vitamin equals greater than biologically active. Vitamin Z and K. Oxidation reduction cofactors, lipid quinones. Vitamin E, structure. Substituted aromatic ring along isoprenoid chain, asterisk ring HC, asterisk isoprene subunits, berry. Hydrophobic, asterisk associated with membranes, asterisk associated with lipids dissolved in blood and lipid deposits. Vitamin E, function. Biological antioxidants react with free radicals, I, superoxide, prevent oxidative damage to unsaturated Fa and membrane lipids, asterisk can cause them to be fragile. Vitamin K undergoes oxidation reduction RxN during prothrombin formation, asterisk enzyme that produces fibrin from fibrinogen, asterisk promotes clot formation, found in green plant leaves, ring hydrocarbon, isoprene subunits. Lab analysis of lipids. 1. Lipids are unlike proteins in physical properties, asterisk organic, nonpolar solvents, asterisk not aqueous solutions. 2. In general, separation of mixtures of lipids utilizes a. differences in polarity, asterisk differences in solubility in nonpolar solvent, lipid extraction in organic solvents. Neutral lipids, ethyl ether, chloroform, benzene, membrane lipids, polar organic solvents. I.e., ethanol or methanol solvents reduce weak interactions, i.e., changing bonds change structure. Lipid extraction method. Phase extraction, chloroform 1, methanol 2, water 0.8. This is one phase and extracts all lipids. Add more water equals greater than separates two phases, A, water, methanol phase on top with proteins, sugars, B, organic phase on bottom with lipids. Adsorption chromatograph. Separation of lipids based on polarity, column chromatography, thin layer chromatograph. Two-dimensional thin layer chromatography, gas liquid chromatography. Column chromatography. 1. Column packed with insoluble polar material. 2. Sample added, polar lipids bind to column. Neutral lipids don't. 3. Column washed with solvents of increasing polarity. Neutral lip are diluted. In first wash, chloroform, other lipids eluted using solvents of increasing polarity. Very polar or charged lipids eluted with alcohols. Thin layer chromatography. Tilde column chromatography, but asterisk plate of glass coated with silica, asterisk sample applied as spot or line to bottom of plate, asterisk bottom of plate up to spot, placed into solvent, solvent rises into silica through capillary action, asterisk lipids separate, lower lipid polarity, more lipid moves not bind. as tightly. Lipids detected with iodine vapors, fluorescent molecules, or specific dyes. Two-dimensional thin layer chromatograph. Separate, not identify. First time, get bands, dots. Each band can be multiple lipids. Second time, turn 90 and put in new solvent system. Go again. Gas liquid chromatography. Separates charged lipids best, based on 1. Solubility, 2. Volatility and solvent, goes from liquid to gas states with increased temperature, many lipids naturally volatile. Gas liquid chromatography method. 1. Add sample, derivatized sample into column packed with inert material. 2. Heat column to volatilize lipids to be separated. 3. Force volatilized lipids through column using inert gas, helium. 4. Order of elution depends on a. Nature of solid column support, b. BIP of lipid, or derivative need. Mass spectrophotometry to identify